So, I'm going to start off with just some fun laser speckle physics today, and how we use laser speckle to measure blood flow across length scales from the human down into invasive measurements in the animal brain. But then over tomorrow and the next day, we'll be talking more and more about brain function and actually how we use optical methods to better understand the fMRI bold signal of brain activity. Okay, so let's get started. So as I just said, today I'm going to tell you about laser speckle dynamics and how we measure that in humans and non-invasively and invasively in animal models. So actually, when I started my PhD, I actually did my PhD with Arjun Yode, who just left 2 days ago. I went into the lab and he was studying colloidal systems of microspheres in solution, and he was using light scattering technology, and he asked me if I would use this technology and make it biological. And so he introduced me to Britton Chance, where instead of studying colloidal systems, I used fish tanks of essentially milk to mimic tissue. And so we were trying to develop a diffuse optical tomography to measure actually breast cancer at the time. But I came back and I wanted to measure not just hemoglobin by absorption spectroscopy. And in this experiment, this is a classic experiment. Arjun probably told you about it. If you put a near-infrared spectroscopy system on your forearm and you have 2 or more wavelengths of light, you can measure the hemoglobin content because oxy and deoxyhemoglobin absorb light differentially. And if you do a pressure cuff and occlude blood flow, you can actually see the deoxygenation of the blood because you're no longer delivering oxygenated blood to your arm, right? So what we wanted to do was instead use a coherent light source that would have a speckle pattern and measure the speckle dynamics and see if we could see a decrease in blood flow. And so actually this was the first experiment we ever made with diffuse correlation spectroscopy. It was actually me with Igor Maglinsky, who was a student actually with Valery Tuchin, who he sent to Philadelphia in 1995, and I had a great time working with him for a year. And this was the first measurement using diffuse correlation spectroscopy to measure the change in blood flow. And as you can see, when we increased the pressure in the arm and included blood flow, basically this blood flow dropped to 0. And as we released the pressure, blood flow slowly came back and back, had a hyperemic overshoot and returned back to baseline. Okay, so if anyone wants to know the details of the physics of how we actually go from measuring speckle fluctuations to quantifying blood flow, I'd be happy to delve into the details. Right now, I'm just going to kind of give you a brief overview summary of the theory. But before going into the theory of diffuse correlation spectroscopy, it's important actually to jump back almost 50 years to dynamic light scattering, which was actually developed at the beginning, the advent of lasers, right? So it's much simpler to actually think about light scattering once from a suspension of particles. So in this case, imagine a simple scenario where you have an incident laser beam and a suspension of particles. You're measuring light scattered at a specific angle, so you can measure that angle. Of course, you're controlling that. And if these particles are moving, it's going to impart a Doppler shift on the light. And so actually what you're going to measure at the detector are all of these scattered waves that have slightly different frequencies because of the Doppler shifts they got from the moving particles, right? And so that will actually produce a beat frequency at the detector, and essentially what you're going to be measuring is an intensity fluctuation that's arising because of speckle fluctuations that are arising because of the Doppler shifts imparted on the light by the moving particles, right? So we can actually, we measure intensity, we can measure the power spectrum of the intensity fluctuations, or we can measure an intensity temporal autocorrelation function. This is something we like to do for 2 reasons. One, when we're making our measurements, we're usually photon counting because we have to measure a single speckle of light. To measure a single speckle of light, we need a photodetector aperture, generally of about 5 microns. Typically with like a near-infrared spectroscopy measurement, you might have a millimeter aperture. So 5 microns is 200 times smaller diameter-wise, which is 40,000 times smaller area-wise. So you collect a lot less photons. And so we like the photon count. And so when you're photon counting, it's relatively straightforward to calculate the intensity autocorrelation function. The other reason I like the intensity autocorrelation function is we can theoretically relate that to the motion of the scattering particles, right? So fortunately, you know, this power spectrum and the autocorrelation function are well related by the Wiener-Kitchum theorem. But let's just focus really on the intensity autocorrelation function. This is actually related to the field autocorrelation function by the Siegert relation. The field autocorrelation function is generally harder to measure. You need to measure actually, you have to use an interferometer to measure the electromagnetic field that is being scattered from the particles. Generally, we just measure the intensity directly. So it's very nice that we have this relationship because theoretically we can derive this field autocorrelation function G1. But experimentally, it's much easier to measure the intensity autocorrelation function. Right? So this is the normalized field autocorrelation function. And again, if anyone wants, I'd be happy to go through the details of these derivations. But this is just related to the scattering angle of the light, the momentum transfer Q, and the displacement of the scattering particles. Right? So we could average over all the ensemble displacements of the scattering particles, and we get this very simple relation that gives us the decay of the autocorrelation function, which is dependent on the mean square displacement of the scattering particles. So if you have a suspension of scattering particles that are undergoing Brownian diffusion, then basically your field autocorrelation function is going to decay as the Brownian diffusion coefficient times time, right? So very simple exponential decay. And very early on, shortly in the 70s, shortly after the invention of the laser, this was being used actually for particle size distributions. People had chemical reactions and particles in suspension, and they wanted a simple question: what is the size distribution of the scattering particles? And they could very simply use dynamic light scattering and measure the intensity fluctuations, measure the decay of the intensity autocorrelation function, and from that decay rate, they can actually get the diffusion coefficient, which was dependent on temperature, the Boltzmann constant, and the radius of the particle, right? So this is a very simple way to get distributions of the particle sizes that you would have in a suspension that scattered light, okay? Now, shortly after that, in 1975, actually, was the demonstration that you could use this laser Doppler dynamic light scattering method, actually, and measure blood flow in tissue, right? So laser Doppler blood flowmetry. And so it took a simple laser Doppler device that was being used for particle size measurements, just put it on a fingertip, measured the power spectrum. Remember I said power spectrum and intensity autocorrelation function are just Fourier transform pairs of each other. And then this is the power spectrum on the finger, and then they just put a rubber band around the finger to stop blood flow to the fingertip, and you saw the power spectrum reduced, right? So that means there's less blood flow, there's less Speckle fluctuations, right? Okay, so that's cool. Now, let's take this to diffuse correlation spectroscopy. So diffuse correlation spectroscopy is the exact same thing as this dynamic light scattering, but instead of scattering once from the medium, we're measuring photons that have scattered typically hundreds or thousands of times, right? And so in our theory, we now have to consider the sequence of all of those scattering events. Mathematically, fortunately, each scattering event is pretty much uncorrelated with the previous scattering event, and so this really just reduces to the dynamic light scattering problem raised to the nth power, where n is the number of scattering events. And so we can go through that mathematical derivation, and kind of the key point of all of this is that it's what we measure with diffuse light that it has highly scattered within tissue is pretty much the same thing you measure with dynamic light scattering, single scattering, with one difference. With the diffuse light, we actually have to convolve over the distribution of path lengths through the tissue, and when we do that, we see that what we're sensitive to is the square root of the mean square displacement of the scattering particles, whereas with single scattered light, you're just sensitive to the mean square displacement of the scattering particles, right? So very simple, it's just a square root difference. And we're sensitive to the mean square displacement of the scattering particles. So if it's Brownian motion, it's the diffusion coefficient. And if it's an ordered convective motion, like with blood flow, we're sensitive to the speed of the moving particles. In tissue, it would be red blood cells, right? So going back again to the very first measurements that actually Igor Miglinski and I made on our forearm. This is the diagram. We had a cuff, and remember we saw this drop in blood flow. And to get this blood flow index, what we did was we actually measured the intensity autocorrelation function, and we looked at the exponential decay of this correlation function, and we essentially got the slope of that. And so essentially here what we're plotting is the slope and relative changes in the slope, which is an indication of the relative changes in blood flow. Now, at the time, to get that exponential decay, we realized it had to be the square root of tau. Kind of initially, I thought it should go as tau, because it would be the square root of the mean square displacement. The mean square displacement of moving red blood cells, that would be speed. That should go as tau. But the data was showing the square root of tau. I was close to graduating. I didn't think deeply about this. I said, okay, let's just use square root of tau. I'm happy with that. But it was really bizarre, because that meant what we were measuring was the diffusion of red blood cells, not the linear motion of red blood cells, right? So we never really understood that. I got my PhD, fortunately, and many years proceeded, where Arjun, and as I'm sure he described to you, they spent over a decade doing extensive validation of the methodology against gold standard measurements. And so even though we didn't understand why we were measuring this random, unordered motion of red blood cells, it still proved to actually be a very useful measurement of blood flow as validated against many gold standard methods, right? So in Boston, I was working with Mariangela Franceschini, and she was working with infants, and so I suggested to her she might want to complement her near-infrared spectroscopy measurements of hemoglobin in infants by adding this diffuse correlation spectroscopy to measure blood flow. And what's really cool is if you combine, I'm sure Arjun was telling you about this, if you combine measurements of blood flow with measurements of oxygenation, you get a measurement of the metabolism of oxygen in the tissue, right? So that turns out to be clinically a very useful parameter. So Maria Angela took this into the clinic, has made measurements on hundreds of babies. I'll just tell you some of her results, looking at infants who are brain injured at birth, unfortunately. This sometimes happens. This is an infant that actually had a stroke at birth, and on MRI you can see the hemisphere that was deprived of oxygen. This is in the first week, and by measuring hemoglobin, you can see in the healthy hemisphere what is a very typical oxygen saturation of the hemoglobin, and this is this cerebral metabolic rate of oxygen relative to healthy controls. It's actually somewhat elevated. If we look at the injured cortex, we see a very high oxygenation, which you might expect if you think the tissue is dead, it's not consuming oxygen. And if it's not consuming oxygen, hemoglobin oxygenation will be high. But in fact, when we measure blood flow, we actually see that the consumption of oxygen is very high, right? So we were very confused about this at the beginning, but what we realized is in that first week of injury, the brain is actually experiencing seizures. The tissue is not yet dead, it's in the process of dying. And in the process of dying, the brain tissue is seizing and it's metabolically very active, right? And this high metabolism is actually toxic to the tissue and it leads to death, right? So we were able to measure that high metabolism, whereas if you just measured oxygenation, you might think the tissue is already dead. But this means the tissue is alive, but it's in the process of dying. You measure several weeks later, and you actually see that the metabolism has essentially gone to zero because the tissue is now dead, right? But the oxygenation looks normal. That's very curious. It suggests the tissue is fine, but actually the tissue is dead. So they've now in Boston and most cities around the world, infants who are born with injuries, and they're given like anti-epileptic therapy, or their brains are cooled to try to reduce that seizure activity. And the purpose is to reduce the metabolic damage that's happening to the brain. And so she has now measured that actually under this hypothermic therapy, the metabolism is actually reduced below normal, right? So that's what the therapy is supposed to do. And now you can imagine using this as a guide for how many days the therapy should be needed, and then when you bring them off the hypothermic therapy, and hopefully you want to see the brain go back to normal metabolism and not hyper increased metabolism. Okay, so that's just a snapshot of potential clinical utility. Let's come back to the question of why are we measuring diffusive motion, right? So 10, 15 years has passed. We've been using this clinically for many interesting studies, but we still didn't know why we're measuring diffusive motion. So then I came across some papers that taught me two things. One, the scattering length of light in blood is about 10 microns. 20 microns is very short, right? So much shorter than I had imagined. And so what that told me is, well, light's scattering multiple times in a blood vessel. This may make me sensitive to the relative movement of the red blood cells. And the other bit of information I found was that actually the red blood cells under shear actually create a hydrodynamic jostling of each other that kind of causes a random motion of the red blood cells. So the red blood cells are undergoing random motion, and I have multiple scattering, so maybe I'm more sensitive to the relative displacement of the red blood cells and not that linear bulk movement of the red blood cells, right? So that led me to think we were sensitive to this diffusion, this hydrodynamic diffusion of the red blood cells. And so we went back to a lot of this infant data that Mariangela had. We fit this equation to the data. We got a better fit to the data. So we thought that this was good evidence and an explanation of why we were sensitive to this diffusion of red blood cells. It was actually the shear-induced diffusion of red blood cells that we were sensitive to. So I went back to Arjun, who was my PhD advisor. I told him this stuff because he too had been wondering for over 15 years, why are we sensitive to the diffusion? He said, well, this is a nice idea, but it doesn't prove anything. And he was a good physicist. It was just a conjecture. I didn't actually provide any proof. So the only other thing I could do at the moment was actually run Monte Carlo simulations, something I like to do. You can very easily kind of on a computer simulate how photons are scattering through a highly scattering medium. So we created this heterogeneous medium where we had blood vessels running through the medium in an ordered fashion, and we're recording the positions of all of the scattering events. And importantly, we track when a photon would enter a blood vessel and exactly how many times it scatters within the blood vessel and the scattering angle. And when we have all that information, we can then really calculate what is the effect of the linear motion of the red blood cells on the correlation function versus what is the effect of the shear-induced diffusion of the red blood cells on the decay of the correlation function. And kind of, I'm going to take you a few slides through this, but the short answer is we got it half right. We are, in fact, more sensitive to the shear-induced diffusion of red blood cells than we are sensitive to the linear ordered motion of the red blood cells. But we don't actually need multiple scattering within the blood vessel, right? So that was interesting for me to figure that out. So we ran lots of simulations. The first was actually, there's the scattering length. Light scatters on a very short length scale in tissue, but when it's scattered in a highly forward direction. So it has to scatter many, many times before its angle is randomized. And typically when we measure through a centimeter of tissue, what we care about is not the scattering length, but what we care about is that random walk length. So in tissue, you might typically say the scattering length is 100 microns with near-infrared light, but that random walk length is 1 millimeter. That's the distance over which a photon's angle is randomized. And so we wanted to ask the question, with these small red blood cells, blood vessels, where certainly light is not diffusing in a blood vessel, are we more sensitive to the scattering coefficient or to the random walk length, the reduced scattering coefficient? And so we ran simulations keeping actually the reduced scattering coefficient the same, but changing scattering and changing G, which is the scattering anisotropy function. And then, I mean, this is the formula. Once we collect all those photons, we can calculate the correlation function, and we can actually turn off the shear-induced diffusion, or we could turn off the linear motion of the red blood cells, right? And so this allows us to then answer the question, what causes a faster decay of the correlation function? Is it the diffusion or the linear motion of the red blood cells, right? So when we do that, we see that if we have no diffusion, it's just the linear convective or ordered motion of the red blood cells, the decay is slower by an order of magnitude slower than if we look at the diffusion, right? So that was our first, kind of microscopic evidence that we're sensitive to the shear-induced diffusion of the red blood cells, right? And it was basically independent of the scattering coefficient. It was only dependent on the reduced scattering coefficient, right? So this basically said we don't need multiple scattering within the blood vessel to have this effect, right? We then looked at, we could change blood flow either by changing the speed of red blood cells or by changing the diameter of the blood vessel. And so here we're looking at what happens as we change the speed, and basically we see that the slope is linearly increasing with speed. This then allows us to relate the blood flow index with the diffusion coefficient, and the proportionality is actually this probability of scattering from a red blood cell, right? Now the probability of scattering from a red blood cell is going to depend on the blood volume fraction, right? So that's going to depend on the diameter of the blood vessel. So that was the next thing we looked at. We changed blood flow by changing the diameter of the blood vessels, but keeping the speed of the red blood cells constant. And so in this case, that blood flow index increases with blood flow, but it has this nonlinear dependence, right? And I won't go through all the math, but basically, at the end of the day, after running all of these simulations, we see that this blood flow index we measure with diffuse correlation spectroscopy is linearly proportional to the absolute blood flow in tissue, so we're very happy about that. But it's modulated by actually a factor which depends on the radius of the blood vessels you're measuring, as well as the probability of scattering from a red blood cell, which really it depends on the scattering coefficient of blood versus the scattering coefficient of the tissue, right? Which is also a function of the hematocrit, the blood volume fraction of hemoglobin, right? So there is a dependence on hematocrit, and it's a nice linear dependence, so it's nice to see that. Okay, so the end of all of this is that diffuse correlation spectroscopy is linearly proportional to absolute blood flow, but it's modulated by the vessel radius and the hematocrit. And we don't actually need multiple scattering within a blood vessel, right? We did notice this trend that as we made measurements at shorter and shorter length scales, source detector separations, that the ordered motion of the red blood cells would become more important. It was still the disordered motion that dominated. So it suggested to us that maybe with laser speckle contrast imaging, which I'm about to tell you about, we might actually be more sensitive to that ordered convective motion rather than the disordered shear-induced diffusion of red blood cells, right? But that needs further investigation. So I told Arjun about these results. He said, well, that's interesting, but it's a Monte Carlo simulation. I don't really care about that. You need experimental proof. So, I always wanted to please my advisor, even though I was a professor now of my own. I still needed to please my PhD advisor. So I was very depressed because I didn't know what to do at this point. So I gave up for a while. So let me tell you about some other things I was doing. Laser speckle contrast measurements. I played with for a while, had a lot of fun playing with it with Andy Dunn when he was at Mass General, and he and I were working together. We were just very fortunate to be able to kind of apply it for the first time to study the brain. Well, laser speckle contrast, I mean, as Arjun probably told you, right, if you just illuminate a piece of paper with incoherent light, might look like this, but if you use laser light, coherent light, you're going to see a speckle pattern, right? So that's just the constructive interference of the light that is being scattered from that rough surface, right? So tissue is essentially a rough surface. You're detecting photons that have traveled multiple path lengths, and so it's going to interfere constructively or destructively, right? So when the surface is moving, or say there's red blood cells moving in the tissue, it's going to change the phase of the light in a time-varying fashion, and that speckle is going to fluctuate in time, right? And so we can actually measure speckle contrast. You can do that spatially. You can just take a small region, measure a speckle contrast. That would be just the standard deviation of the intensity divided by the mean. That's speckle contrast. And so this is a measure of the spatial statistics of the speckles. Right? So if we have a camera exposure of the brain, right, and say that exposure of the surface of the brain is longer than the time scale of the speckle fluctuations, then what's going to happen is the speckles are going to blur out, right? Because there's temporal fluctuations. We're blurring that with the camera picture. And so this is a picture of the surface of the brain with near-infrared light, 800 nanometer light. And actually, you can kind of see these big blood vessels, and you're seeing them not because of the absorption of the blood. This is not an intensity artifact. It is slightly. But mostly what you're seeing is actually differences in the spatial statistics of the speckle pattern, right? So here there's high spatial fluctuation, speckle fluctuations, and here the standard deviation is small, right? And it's small there because there's high blood flow in that blood vessel. So if I just go in and measure the speckle spatial statistics at every local point, you get this image, right? It's the same data, it's just the speckle statistics. This always amazes me. So this is speckle contrast, right? So white, there's about 20% standard deviation of the speckle intensity relative to the mean, and black is essentially approaching 0 standard deviation, right? The speckles have been blurred because of the speckle fluctuations over the integration timescale of the camera, right? It's amazing. You can even see all of these very small vessels that we could not see in the intensity measurements over here. Okay, so cool. I think I'm going to skip all of this derivation. We've done validation against laser Doppler blood flowmetry. So this was, yes? Yeah, so typically for all these measurements, you want the coherence length of the light source to be longer than the distribution of path lengths through your sample. All right, so that if as long as that's the case, then all of the photons that you're detecting are going to be interfering with each other. Right? So for a measurement like this, the distribution of path lengths through the sample is going to be 1 or 2 millimeters. And so we typically use laser diodes. Although that can be a problem because laser diodes can have coherence lengths, say, of 100 microns, less than a millimeter. And that reduction in coherence length will reduce your speckle contrast, right? So you do have to pay attention to that. For the diffuse correlation spectroscopy measurements, when we're making measurements through centimeters of tissue, we like to use lasers with coherence lengths of 1 meter or longer, right? Just because the distribution of path lengths through the tissue can be, tens of centimeters, right? Okay, so it was interesting to me when we wanted to publish this paper. We were working with a neuroscientist, Mike Moskowitz, who studies stroke, and he said we needed to validate against laser Doppler blood flowmetry. I said, well, the physics is exactly the same. Why are you making me validate this? He said, well, because everyone in my community uses laser Doppler blood flowmetry. They have no idea what laser speckle contrast imaging is. I said, the physics is the same. He said, but we have no idea. So he made us do this validation measurement, and of course we got a perfect correlation. And so that went in the paper and made my colleagues happy. So then, we validated that. Now, Mike Moskowitz wanted to use it for problems he was interested in. So one thing he was interested in was cortical spreading depression and stroke and migraine headaches. Cortical spreading depression actually is, is a wave of depolarization that spreads across the surface of the brain. It actually, anyone who has a migraine headache and has an aura before the migraine, they're actually experiencing a cortical spreading depression. Right? It's just the neurons depolarize and that spreads across the cortex and it creates this aura. Right? So you can measure it electrically. If you have electrodes on the surface of the brain, you can see this depolarization, and it takes a few minutes to move a millimeter. Right? Now, when the neurons depolarize, they have to repolarize. So the pumps have to increase their activity to pump more ions across the membrane. To do that, they need more ATP. To get more ATP, they need more oxygen. To get more oxygen, they need more blood flow. Right? So we could measure blood flow and essentially measure this propagation of a cortical spreading depression across the surface of the brain. Now, it's interesting, to create a cortical spreading depression, you can do one of two things. You can either drop potassium, potassium chloride on the surface of the brain, and that extracellular potassium will actually induce this spreading depression, or you could poke the brain with a pin, and it will do the same thing. And so I forget which we did in this case, either the potassium chloride or poking the brain with a pin. And this is marching in 10-second intervals, and red is an increase in blood flow. And you can see blood flow increases propagating across the surface of the brain, right? So this is corresponding exactly with that depolarization of the neurons and their repolarization, right? Okay. So this also happens in stroke. I wish I had the movie, but I don't have the movies. So if you have a stroke, you have a core where you have blood flow reduced to below 20% of the baseline value. When that happens, the pumps aren't able to maintain the ionic balance of the neurons. You get a buildup of extracellular potassium, and when that extracellular potassium goes above a certain threshold, it causes the pumps to the channels to open up, and the intracellular potassium will flood out. And so you actually get this wave of extracellular potassium diffusing across the brain, and so that will actually happen on the edge of the core of the stroke, where you have this metabolic imbalance and the ionic pumps are struggling to keep the ionic balance. And what happens is you just get generation of spontaneous spreading depression waves. And so, let me show you what happens. So you induce the stroke, you see the core goes down to below 20%. The penumbra is around 40%, and the surrounding healthy tissue is also compromised. But what happens is we see these spreading depression waves happening every tens of minutes. And they actually circle around the core of the stroke. It's kind of interesting. Every time that spreading depression wave happens, you see in the penumbra, it actually, blood flow goes down, but it never goes up. So the neurons are getting a hit. They're depolarizing, but they don't have that increase in blood flow to help them repolarize. They're getting this metabolic insult that's toxic to them, which actually drives them to death faster. And so actually you can see after each spreading depression wave, that baseline blood flow drops, right? And actually, if you look at this, this is the core of the stroke defined as blood flow below 20%. This is before spreading depression. Immediately after spreading depression, you see that the core of the stroke grew, right? So this is tissue that's dead. It's dead. It's never going to survive. Okay, so then the idea was, and this is looking at the area of that core of the stroke, and you can see after every spreading depression wave, the core gets bigger and bigger, right? So it gave my colleagues the idea, if we could reduce the number of spreading depressions, we could reduce the growth of the stroke. And so their idea was, let's give more oxygen to the animal. And so if they have more oxygen, their tissue oxygenation will be higher, the pumps will be able to run better and maintain that ionic balance, and it will reduce the occurrence of spreading depression waves. Okay? And so that was their idea. This is normal oxia, normal oxygen, and you see these spreading depressions. In an animal where they put on 100% oxygen, you see that spreading depressions essentially disappear. Right? So this is one animal. If you do the statistics over many, many animals, you basically see the spreading depressions go away. So what's the impact on the size of the stroke? You can basically see, in a normal animal, the stroke continues to grow over time. but when you give this 100% oxygen, the stroke is no longer growing in size, right? So that seemed really good, and something we should do in humans. And actually, there have been studies investigating that, but it turns out humans are more complicated than rats, so there's other factors going on as well that need to be controlled for. Okay, so this is cool, but we still don't know laser speckle contrast imaging. Is it measuring the convective ordered motion of red blood cells, or is it measuring this diffusive unordered motion, right? So we couldn't yet answer that question. So I started to develop other techniques. This laser speckle contrast imaging is just measuring blood flow on the surface of the brain, and kind of, it's not resolving in depth. But we could use optical coherence tomography to resolve blood flow in the brain. I think you haven't heard about optical coherence tomography yet, but I think you will in a few days, in course 11, I think. But it's basically, it's like ultrasound, but instead of using sound waves, we're using light waves. So you could think of a pulse of light going into the tissue, and you're measuring the reflections of the pulses of light coming out. We do this interferometrically. So you can resolve with great resolution in depth the signals coming back. And whenever there's a moving red blood cell, it imparts a Doppler shift, and we can measure that Doppler shift. And also, because we're doing this interferometrically, we actually measure the field directly, and we can measure the field temporal autocorrelation function and relate that to the blood flow, right? So, this is what an image in depth would look like in the brain. You don't see anything interesting, but if you look at the Doppler shifts, you can pick out moving red blood cells, and if we sweep this over a volume, we get just incredible images of the blood vessels, right? And so we can actually go in and measure the speed of the red blood cells in all of those vessels, right? So very powerful for a number of applications. And so I was lucky enough to work with postdoc Vivek Srinivasan, who did his PhD actually with Jim Fujimoto. And so he came to my lab and helped us kind of adopt this for the first time to measure blood flow in the brain. And it can do many interesting things, right? So it resolves in 3 dimensions, blood flow at a microscopic level. We can quantify the blood flow because of Doppler. We can measure the passage of red blood cells in capillaries. And we can even measure intercellular motility. That I'll show you a little bit of that, right? So to do that, we want to do dynamic light scattering with OCT. So typical dynamic light scattering, right? You have a laser beam shining on a test tube, suspension of some scattering particles. You measure the single scattering at some angle, and you can quantify the motion of the scattering particles. But in tissue, you have multiple scattering. It's very hard to control where that scattered light is coming from. But with OCT, because we're using this interferometric measurement and a coherence gate, we can coherently gate exactly where the photons are coming from, at least what path length of photons, right? So that essentially allows us to control very precisely the measurement of singly scattered light within tissue, right? And so we could derive the dynamic light scattering theory, but for OCT. If you're interested, I'd be happy to talk to you afterwards about how you derive this. But it predicts very nice the field autocorrelation function. It's got 3 main components. So we make measurements every tens of microseconds, and in tens of microseconds, 3 things can happen. A scattering particle could move in or out of the voxel that we're measuring. There could be scattering particles that are not moving, and there could be scattering particles that are either diffusing or flowing through the voxel, right, on a time scale that's slow compared to our sampling time. So when we measure this, we're going to measure a field autocorrelation function that will look something like this. This is the stochastic component. Here's a flowing component, and then the amplitude decays because of decorrelation that happens because of random motion of the red blood cells or red blood cells moving in and out of the voxel. All right. So we can apply this to measurements in the brain, and what's nice is we can quantify the speed of the red blood cells and the diffusion of the red blood cells. And we can do this on, so if we zoom in here, you can look at that point and measure a field correlation function that looks exactly like a theoretical prediction. You can do that at different locations, and we're fitting the field correlation function, right, to get the speed and the diffusion coefficient, right? So we can do this on different length scales, and everything works out quite nicely. And one thing that kept showing up was this yellow. This yellow is actually diffusion of red blood cells, right? At the time, I thought this was an artifact. Like, why are we measuring diffusion? It must be that, and it was always on the edge of the blood vessels. And I thought, okay, well, maybe the blood vessels are kind of oscillating. There's vasomotion of the walls of the blood vessels, and my model is not accounting for that. So this must be a model error. That's what we thought, right? So I just want to show you briefly, if we look outside of the blood vessels in the tissue, we saw these spots that had diffusion, but no motion of red blood cells. This turns out this is actually the motility of intracellular organelles that are diffusing within the cell body, right? So usually measurements like this are only made in vitro in cells, and never made in vivo because you can never isolate the signal just from a cell body, right? But with OCT, we can do that. So this is really cool that we were able to do that, and I think it will have interesting applications in other arenas. But let me just come back to the blood flow problem. All right, so just to recap, in 1995, we used diffuse correlation spectroscopy to measure blood flow in tissue. We measured diffusive motion. We didn't know why it was diffusive. We were expecting a linear motion of the red blood cells, right? So we came up with this idea that we're actually sensitive to shear-induced diffusion of the red blood cells. Arjun thought it was a nice idea, but it didn't prove anything. So we ran Monte Carlo simulations that supported this idea. Arjun wanted experimental proof, right? So we didn't have experimental proof, right? And so I'm not neurotic. It's not just that I want, my advisor to like me. It's that it's important. I think clinically, it's a really important tool for measuring blood flow, and it's important that we understand what we're actually measuring, right? But we need experimental support for this. And then it occurred to me, shoot, I just showed you OCT was measuring this diffusion on the vessel wall, which I didn't understand at the time, but actually the diffusion is happening near the vessel wall. It's not on I thought it was on the vessel wall, but it's actually near the vessel wall. Near the vessel wall is where the shear is greatest. And where the shear is greatest is where the shear-induced diffusion of red blood cells will be the greatest. So I had a new postdoc arrive in the lab, Jumbo Tang, and so I asked him, could you just go in and please measure the shear-induced diffusion of red blood cells? And let's just see if actually this relation between the linear motion of the red blood cells and the diffusion is predicted exactly by the shear, right? So he went and did that. So he made measurements on many, many mice, and he'd find these penetrating vessels where we could see very nicely in the center. I think I blow that up. Well, you can see red in the center is where it's just linear motion, and on the edge surrounding it is the diffusion, right? So here we're looking at the correlation function, and here's just kind of raw data. And you see the correlation function is decaying more slowly in the center of the vessel and faster near the edge of the vessel, right? So I had always just conceptually thought, in the center of the vessel, that's where the linear motion of the red blood cells is the fastest, right? Because there's a laminar speed profile. And where the linear motion is the fastest, that's where the decay of the correlation function would be faster. This is what I conceptually thought. But actually, now that we measure with OCT, we actually see the correlation function decays faster near the edge of the vessel wall, which is weird, because the edge of the vessel wall, that's where the speed of the red blood cells goes to 0, right? So the speed is slower, so the correlation function should decay more slowly. But it's decaying faster. So the only way it could decay faster is because of the shear-induced diffusion of the red blood cells, right? And so Jumbo measured that. So basically, he's able to measure the laminar profile of the speed and the laminar profile of the diffusion coefficient, right? And so we expected a linear relationship between shear and diffusion. Shear is just the radial gradient of the laminar speed profile, right? And so basically you could see that the model fits the data very well. It gives you the proportionality constant, this alpha shear, and he did it for different size vessels, and some very interesting things come out of the data. One, this laminar profile is a plug profile. You know, in large vessels, it is purely laminar. It's a quadratic relationship with the radial distance from the center. But in smaller and smaller vessels, it becomes plug flow. And it's no longer quadratic. It actually becomes cubic or maybe even to the fourth power, right? So we can measure K, which is this plug parameter, and alpha shear versus vessel diameter. And we see that for large vessels, we get laminar profile, but for smaller and smaller diameters, it becomes more of a plug flow. And this alpha shear, we were measuring about 0.2 millimeter, 10 to the minus 6 millimeter squared. Ex vivo measurements were done in the late 1970s. These would be with ghost blood cells moving through a glass capillary tubing. So that shear-induced diffusion was all characterized in the late 70s ex vivo, right? And they measured parameters that were about a factor of 5 larger than what we measured, but we were for the first time measuring it in vivo. So we were happy that it was within a factor of 5 of those old measurements. So I was all excited. I thought I finally had the experimental proof to make Arjun happy. And so I saw him March 19th at a dinner in Washington, D.C., and I told him about it, and he said, well done, but what happens if alpha shear, when blood flow increases, right? And so we didn't report that, but fortunately it was in our data, so I was able to go home the next day and talk to Jumbo, and he was able to make this graph, which basically shows, as for different size diameters, actually, for low blood flow, alpha shear is high, and as blood flow increases, alpha shear decreases, right? So it's important to understand this biophysics. So ultimately, what we're measuring with diffuse correlation spectroscopy is the shear-induced diffusion. We're not actually measuring blood flow directly, right? So we need to understand what this proportionality constant is between shear and blood flow. Okay. I still didn't answer the question, what are we measuring with laser speckle contrast imaging, right? So with the OCT, we got that experimental evidence that shear-induced diffusion is really important, but it doesn't tell us if it's important for these sorts of measurements, right? Now, fortunately, Dmitry Posnov arrived in the lab last year, and he wanted to delve very deeply into laser speckle contrast imaging. He's very, very good at that, and he's made very good progress on this. So just to recap, so the correlation function has 3 different forms. It can decay as tau, it can decay as the square root of tau, or it can decay as tau squared, right? And these are all related to basically how much light scattering there is and whether or not there's ordered or unordered motion. For laser speckle contrast imaging, almost universally, and laser Doppler blood flowmetry, universally people use this exponential model, tau, right? Which is either multiple scattering of light from the ordered motion of moving red blood cells, or it's single scattering from unordered motion of red blood cells. Okay, people just use this. As Arjun would say, they don't really have experimental support for why they use it, right? But Dmitry, he found a camera that would allow him to measure. So laser speckle contrast imaging, we usually integrate for a few milliseconds, which is much longer than the time scale of the speckle fluctuations. So we can't rarely measure this decay fast enough, right, to distinguish which model is accurate. But if we had a camera that could go much faster, we could do that. So there are now consumer-grade cameras, $5,000, that allow us to get 25,000 images per second. And so he bought that camera, and he started making measurements. Now, we don't get the whole image at 25,000 frames per second. We have to break this up into like 10 images. In each subdomain, we get 25,000 images per second. We can measure the intensity autocorrelation function, how it decays. We can now look at an artery, a vein, and parenchymal blood vessels, and we can actually directly measure the decay of the intensity autocorrelation function, and then fit this model. Does it go as t squared, t, or the square root of t, right? And when he does that, this is what he finds, right? So green is the model everyone's using to analyze their data, right? It goes as t. Red is t squared. So red, basically, t squared is only happening in the largest vessels. So t squared is where we measure light that is scattered once from a moving red blood cell, and it's ordered linear motion. Green is either multiple scattering from ordered motion or single scattering from unordered motion, right? But that only happens in a very small number of regions and in these vessels here. Everywhere else, actually, we're seeing the square root of tau behavior in decay in the intensity correlation function, which nobody ever imagined for laser speckle contrast imaging or laser Doppler blood flowmetry, but it's the majority of the locations, right? So this is interesting. It's just like DCS, right? And it changes basically 20 years of laser speckle measurements of blood flow. You have to reanalyze the data because you have to use the square root of tau model. Now, it turns out this is not so important for the vast majority of changes, because when you only have small changes in blood flow, you're not actually sensitive to these differences. But if you have big changes in blood flow, you become really sensitive to these different model differences. Now, a curious thing is, in these regions between the vessels, we're measuring movement of red blood cells essentially in the smallest blood vessels, in the capillaries. Now, in the capillaries, there's no shear. There's a single file of red blood cells going through the capillaries. So we're not measuring shear-induced diffusion, right? So what are we measuring? Is probably what we're measuring is just shaped fluctuations of the red blood cells as they're squeezing through the capillaries, right? So we need to experimentally prove that. But since there is no shear, that is the only explanation I can come up with. Let me skip. So Dimitri has done lots of cool things analyzing these data. And the last slide, basically the application where this is now relevant is if we look at a stroke with laser speckle contrast imaging. So with OCT, we can see basically during this stroke, there's no blood vessels here. They've disappeared because we only see moving red blood cells. With laser speckle contrast imaging, we measure a reduction in blood flow, in the stroke, we would expect this to be all blue, less than 20% of baseline, right? But we don't measure that, right? That's what we expect. If we use the dynamic light scattering imaging, we do, in fact, measure that blood flow has dropped below 20%, right? So the problem is that we're using the wrong model to analyze the laser speckle contrast imaging. So people have developed this really simple model to analyze the data, right? But it's wrong, and it's wrong for two reasons. It's wrong because it's not using the correct square root of tau. So if we use the correct square root of tau, we see that we get closer to the true decrease in blood flow, but still we're underestimating the blood flow decrease. So the other problem is they simplified and said that 1 over k squared is the change in blood flow, but that's not true. It's much more complicated. You actually need to measure tau c, that time scale of the decorrelation. So if you actually use all of the information and use the proper model with laser speckle contrast imaging, you can get a better approximation of the change in blood flow. Okay, so an interesting lesson from this is that 20 years later, we keep learning more about how to analyze speckle dynamics. And a really curious lesson was when Andy Dunn and I first did laser speckle contrast imaging in 2001, we were not using this simplified model. We were using the proper tau c analysis, and we were properly calibrating our instrument, and we were measuring changes in blood flow in stroke below 20%. I showed you those images, right? But then what happened over the ensuing 15 years was people started simplifying the theory and saying it's good enough, and then there was this creep in the analysis of the data where it was no longer good enough, but people forgot that all the assumptions were breaking down. So that was a really curious lesson for me, was that there's drift to simplicity that gives you worse results. So we have to correct that. Okay, so the take-home message is that laser speckle measurements of blood flow are really cool measurements of blood flow, but it's primarily sensitive to the unordered motion of red blood cells, right? So just, over the last 20 years, I've just had a great group of people to work with, Maria Angela Franceschini, Stefan Karp with the DCS, with laser speckle, contrast imaging, first with Andy Dunn, and most recently with Dmitry Postonov. And with OCT, I've had 3 just great postdocs, Vivek Srinivasan, Jungwon Lee, and then Jumbo Tang do this work. And this was Arjun when I was telling him at dinner in 2017. So if there's time, I'd be happy to answer any questions. All right.
Questions?
Yeah.
Thank you, Doctor, for this wonderful talk. I was thinking about possible applications, and a couple that came to mind was to possibly have a better understanding on this blood flow interaction with cholesterol, with lipids, exactly in the blood vessels, and recently with the CTE that had somehow became more popular research topic. And I don't know if you have some approach on this.
So CTE is the chronic traumatic encephalopathic injury from hitting a hit to head. What does it have to do with lipids?
No, that's a different topic.
Okay.
Having a better understanding of blood flow with lipids and cholesterol, that would be related like to strokes, for example. And on the other hand, the CTE, most of it are related with the blood flow.
Okay, so the CTE is something I think I am very interested in, so both in animal models and in humans, right? So, you get a hit to the head, there's actually a dramatic decrease in blood flow that actually kind of looks like a stroke. And this is kind of scary. So we see this in animal models, but it's very hard to measure it in people because, well, you'd have to get them into an MR scanner or something in a few hours after a hit, and that never happens, right? But with diffuse correlation spectroscopy, right, we can take this to the athletic field, right? And when somebody gets a hit to the head, we can measure it. And I mean, basically, it's like a 50% drop in blood flow, right? So we should be able to measure that on the field and get a measurement of how severe the hit to the head was, right? So that would be a really, really important application. Already in animal models, these technologies are very useful for models of mild traumatic brain injury or concussions, even subconcussive hits to the head. As for the lipids, interesting question, is the lipid content somehow affecting the shear-induced diffusion, right? Good question. It has to be explored.
Thank you.
Yeah.
We have time for one more questions.
Yeah.
Thank you for the great talk. I was wondering in DLS OCT why we were only considering a single scattering of photons inside the blood vessel. So if you can shed more light on that.
Yeah, so you're asking, with OCT, why do we only consider single scattering? Right, so with the OCT, we're basically able to select a path length of light with about 3 micron accuracy, right? So we're only analyzing photons that have traveled a very specific path length of light. From my analyses, we can ignore multiple scattering, right? The effects of multiple scattering. We do have evidence, however, where there is an effect of multiple scattering. Very interestingly, if you have a blood vessel and you measure a path length of light, that would be photons that are coming from underneath the blood vessel, right? Static tissue, and light comes back. We don't measure static, right, tissue. If we go over here, we measure static tissue, but we come here, it's not static. Basically, we're seeing, whoops, you can't see that. So what's happening, the photons going through the blood vessel have scattered in the forward direction. They come here, they get a backward scattering event, but from static tissue. That forward-directed scattering is imparting some Doppler shift on the photons, right? So it is happening, but it's it's a small effect, and so it doesn't adversely impact our analyses inside the blood vessel.
And also the backward scattered photon is due to the change in refractive index between the blood vessel and the tissue?
Yeah. I mean, well, the index, spatial variations in index of refraction gives rise to the optical scattering, and the optical scattering has an angular distribution, right? So it's not strictly backscattering. It has a probability of going in all directions, right?
Okay,
right.
thank you.
Yeah.
So let's thank the professor.
Thanks.